I think the other thing I'd like to add to what Jerry said about me is that I have been working in what could be loosely called soil quality and soil health for the better part of my career, at least directly, for a good part of my tenure here at the Tilth Lab. I was involved with the team that developed and put out the paper with the SMAF, the Soil Management Assessment Framework, and a good part of the past 10 years I've spent working with various teams, and including one of your very own here who was a graduate student here, and there have been papers published on soil health and cropping systems. So you all can go and Google on that. My true love is exactly what Jerry said and where I have the most expertise. So when Jerry asked me to do this at the 11th hour and gave me a topic of C to N ratios, I'm going to go away from the camera for a minute. So I want to hand these things out. Take that. Take a few extra. And I decided I always like to be able to give folks something that's useful to them. And I struggled a little bit because I wasn't really sure y'all would be that enthused about organic. Obviously, that was an assumption on my part. So I went to the internet, and I'm handing out, and I gave you all a copy of this too. Much to my surprise, I found a very nice publication that came out of the Soil Quality Institute, which is now defunct and gone in NRCS. You're what's left of it, I think, the traveling salesman.
Yeah, there's 2 or 3 of us now.
Yeah. Anyway, but this publication probably was put out a decade ago. I want to make sure everybody has it. And it very succinctly and beautifully describes the impact of C to N ratio of crop residues in cropping systems. And I decided that I would hand this out to you and you could read it at your leisure, and I can summarize the high points, and then I can talk to you about where I think that publication can actually be have stuff added to it, okay? Because you can't put a whole lot on 2 pages. But the high points, and I put this up here so that you can have something pretty to look at while I'm talking without slides. Because I like that and appreciate that when I'm in an audience. The take-home message of this publication, which is very, very true, is that the endpoint or the desired endpoint of management, of residue management in crops is to feed the organisms. And it talks a great deal about what the ideal C to N ratio is for your residue inputs on the surface, okay, to feed the organisms. And they talk about a 24-to-1 C to N ratio. Back in the day when I was in school many years ago now, we learned it as 25-to-1, and the point of the 25-to-1 ratio was, and I have, oh gosh, where's the, will this work? Okay. Yeah, just use the down arrow? Okay. Okay, so this is what I learned in soils class many years ago. And it's the same concept. I think that 24-to-1 was used simply because then they could add up the numbers cleanly when they were talking about what part was contributed to what part. And this is the point. If the C to N ratio of the residue that's decomposing on the soil surface is greater than 25, and in the paper they say 24, then the microorganisms will take up nitrogen from the soil because there isn't enough nitrogen present in the residue itself to satisfy their needs for building microbial bodies and for energy. And you will immobilize nitrogen from the soil. If it's less than 25, then the process of mineralization begins to dominate. There is at first just enough, and then finally more than enough N in the residue that they're decomposing and then you start releasing nitrogen into the soil so the plants can take up this nitrogen and for their growth. And that's basically it. And they also talk in the paper about, that little paper, about how much of that 24 is actually used for energy. And so this gives me an opportunity to remember that the organisms are really going after this, the carbon, and going after the organic matter for the energy in the chemical bonds. And they use 16 of those carbon molecules in the 24-to-1. So more than two-thirds or two-thirds of the carbon molecules are used for energy. And what that basically means is that more than two-thirds of it gets lost out the top as CO2. It's called microbial use efficiency. And you've probably heard that. And the remaining carbon molecules are used to build microbe bodies on average, because the C to N ratio of the microbial population of the biomass in the soil is on average 8-to-1. Now, that's on average, and it's simplified so that you can understand it. The truth is that there's a range of microbial types in soil. Soil fungi, for instance, the C to N ratio of soil fungi is on the high end, closer to 10, and the C to N ratio of bacterial bodies is closer to 5, sometimes 4. And so it depends on the mix of the organisms in soil what you're actually going to get. But on average, you can use an 8-to-1 ratio. So none of this is new. As a matter of fact, I almost feel like, you know, it's not that critical to tell you these things, but I think it is, especially after listening to Jerry's talk about the real problems in the world. But this is a situation that the other part of this that we want to focus on in here is that there's a table on the second page that gives you the various C to N ratios of the various crop residues. And this shows you that the narrow C to N ratio materials right up here, like hairy vetch and other legumes, will decompose relatively quickly. And the higher C to N ratios, in this case, this is for cover crop, one cereal rye and another cereal rye. It was just because it was the only picture I could find quickly. Basically, it tells the story. The broader the C to N ratio, the longer it takes to decompose the material. So the end point of this paper is basically they're telling you to reduce your tillage and use extended rotations with legumes and small grains and plant diverse cover crops, all right? That's what they're telling us. That's what they told us more than 10 years ago. And we're finally getting there in terms of you all getting that out in a real way. And the science is catching up with ourselves. Now, what I'd like to do for the remainder of my time is two things. I would like to extend the story to include roots and tell you in a very conceptual way about roots and the interaction of roots and organic matter turnover, something that I've done for a long time. And I'm not going to provide a ton of data. This is basically concept. And then after that, I'm going to talk to you about this organic experiment that yesterday you were slated to have a field trip to see this site, and you didn't go. I'm assuming you didn't go. That's what I was told. So I'm going to provide you an introduction for that and some reasons why we decided to do that site and reasons why I decided to choose working in organic rather than in other systems. So first, this is just a reminder for all of us that soil is also made of a very small part of soil is organic matter, and soil primarily consists of air, water, and mineral particles. So we need to be able to consider the interaction of the soil microorganisms and organic matter with the mineral matrix of the soil. The other purpose for this slide is to re-emphasize the cyclic nature of the transformation of plant residue through the microbial biomass into soil organic matter. All soil organic matter came from plants in one form or another, and it all has to be transformed through the microbial biomass. So the fungi, the actinomycetes, the bacteria, the other organisms in the soil. They're the ones responsible for building organic matter, and when we manage soils, we're managing not only the biomass, but we're also managing the biomass's interaction with the soil mineral particles. So we have abiotic and biotic. And the piece that I want to emphasize here is the importance of roots. For many, many years, we didn't even think about roots in management. And with the increase in no-till during my career, roots have become more and more important in our considerations, but it's very difficult to study roots. And a lot of the data that has been produced on roots has been taken from using a harvest index to calculate or get root-to-shoot ratios and then guess what's in the roots rather than digging them. And these are some really good examples of root pictures. And that's just to show that the part that's important to me is that as the roots grow in the soil, they also exude exudates. And this exudate, this stuff that leaks out in the rhizosphere of the soil, is what feeds the soil organisms. This is very high, very carbon-rich material, very easy for the organisms to absorb and utilize for energy and to build bodies, and that these organisms, in the process of utilizing this material, help to build soil aggregates that stabilize soil structure. And so that's the take-home message from this, that there is an interaction, and that is the interaction. I also don't want to forget that the soil organisms are also, they build their habitat, they build their home through the transformation of organic matter, and they are also responsible for cycling organic matter and for helping to keep the organic matter available to the plants. And this includes critters like nematodes. They live in the pore space. They need water films, or fungi who need 100% aerobic environment to grow. And that's a really nice picture of fungal biomass bridging soil pores taken from the Symbio Center in the United Kingdom. So we've got a need to keep habitat optimized for all of these organisms, as well as feeding them the food that they need. And so that's the idea behind studying soil aggregates and soil structure in connection with the organisms. This is just another picture of a bacterial colony. This is a 5 micrometer scanning electron micrograph to give you an idea of the scale that we're talking about. Very, very small scale where these organisms colonize, and very isolated. They don't occur everywhere. They only occur in patches in the soil. And perhaps Ray talked some about this earlier. So this just summarizes what I just told you. And so very early on in my career, I spent probably about 8 years and a little bit more looking at aggregates and trying to understand how aggregates formed and what sort of organic matter or binding agents were involved in keeping the aggregates intact, macroaggregates, and then also trying to understand how management impacted those processes. And what this came to was, as I was looking for the organic matter that bonded aggregates together many years ago, I was working with a scientist who is now deceased. His name is Ted Elliott. He was my, my advisor for my PhD degree. And what we found is that when we looked at the response of this particular form of organic matter, it's called POM, or particulate organic matter. It's inside aggregates. That this was a very important fraction in land response. This is what it looks like under a light microscope, just little bits of decomposing root residue and pieces of insect carapaces. It's size-defined fraction. It's isolated using a sieve after dispersing the soil. Most of you have probably heard of this. I just want to bring out the data that came out of this that got everyone excited about this fraction, much to my surprise. And this was data that was collected from a field site in Nebraska. And what it shows quite clearly is that in the native prairie soils that were part of this experiment, almost 40% of the soil carbon was held in this particulate fraction. That was very surprising. And that if you tilled the soil, you lost about 50% of it. And if you no-tilled the soil, you were able to start to provide conservation of carbon in this fraction. And, oh, that middle button, okay. Thank you. So we're talking about this line right here. So there is a fairly decent percentage of soil carbon in this fraction, and it responds to management quite nicely. And the other interesting thing that I wanted to bring out here is that the tilled systems, and this is bare fallow wheat tillage in Nebraska. It's dry land, so this is a relatively a system that degrades quite readily. That the tilled system looked quite a bit more like the native prairie in terms of C to N ratio of the particulate organic matter than the no-till. And that really interested me. And so then I looked at what was inside the aggregates. So this is the POM that was inside aggregates rather than floating around in the soil free. And I could see a huge jump in the C to N ratio of the no-till systems. And so what this says is that under no-tillage in these rain-fed dryland systems, we're protecting and saving relatively high quality carbon in aggregates. That's a 60-to-1 C to N ratio. That's pretty close to the C to N ratio of wheat, and maybe even a little higher. Yes?
I guess that low C to N ratio of the prairies kind of messing with me too.
Yeah.
Because the prairies are a very low nitrogen system.
Mm-hmm.
High carbon, high organic matter.
Yeah.
Almost all the native prairie plants are fairly high C to N ratio.
Okay. Well, if you look at the carbon in this one fraction. That's why people got excited about this fraction and why so much work has happened through the past 15 years on this fraction, or more, actually 25 years. A lot of the soil carbon is in POM. The C to N ratio is higher because there's so much carbon in that fraction. There isn't as much nitrogen in POM as there is carbon. And that is different than the tilled soils and agricultural soils. Okay, so the reason why this C to N ratio is higher here is different in native prairie versus tilled soils. Does that make sense?
Why 17?
Why 17?
On the last column.
I would have thought it would have been a lot higher.
Inside? Yeah, that was interesting too. And I don't know the answer to that question.
So was it a prairie that was managed, or was it something that just sat there for a long period of time?
It's virgin.
So
Well,
there
is
was
it
no.
grazing?
There was no grazing, no.
Yeah, that's the problem.
As far as we know, this was native prairie. This is at the Sydney High Plains Research Station in western Nebraska. And as far as we know, this is native land.
But if it hadn't been grazed for a long time, then it's not even gonna be comparable to a true native prairie.
If it hadn't been grazed,
If it hasn't been
well,
grazed in
this,
recent years.
this prairie here, this experiment started in the early 60s, I believe. So from that point on, it was protected from grazing. Prior to that, this is on the CPER.
But that's
So-
the problem, we don't need to protect it from grazing.
That's a whole nother question. See, if I'd have known what you guys wanted to talk about, see, I would have been able to do this a lot better. I hate talking to
So
audiences.
how did the prairies, if they were a very low nitrogen system, very few nitrogen fixing plants on the prairie,
Mhm.
how did they become so productive?
Well-
And why can't we get our ag lands to do that? We think we have to fix all this nitrogen.
Okay, I think the general answer to that is because, very similar to what Jerry was talking about, as a matter of fact, I know that these prairie systems are very much more efficient in their cycling of nutrients and their cycling of water. They evolved over eons. They evolved over, at least the prairies here in Iowa,
Yeah.
10 or 12,000 years. These prairies are much older than that, okay? So they had to come to a place where they came to an equilibrium, where they were producing a certain amount of biomass above and below ground. A lot of the biomass is below ground in prairies, especially in the short grass prairies like we're seeing here, the medium mixed grass prairies, because there's not a lot of rain. And so that gets into a whole nother aspect.
But also animals were part of that.
Yes, the bison were part of that, yes, yes. I also wanted to say one more thing general thing about palm, and this work was done by another group. These are actually biochemists, and they looked at palm, and this is basically palm light fraction, and light fraction is essentially less dense material that is larger and isolated in a different way than palm. But what they're showing here is very interesting. They're showing that the C to N ratio over a broad range of soils is, for this fraction, is between 15 and 25 roughly, and that it's alkyl to orthoalkyl ratio, I believe that stands for ortho, I'm not a chemist. This is a gauge of how humified the material is. Palm looks more like plant inputs than the silt and clay fractions of soil.
So you're saying that
Yeah.
could be more like root exudates, more labile?
This stuff here?
Yes.
Okay, this is particulate or organic matter. I assumed everybody would know about this, so I'm sorry, or I would have spent more time in explaining how this is isolated. This fraction is isolated, so the way it's isolated is it's dispersed with sodium hetamexophosphate, shaken overnight, then the material is poured through a sieve. The basic method in the beginning was a 53 micrometer sieve, so that was done intentionally, sand-sized material, and all the stuff that went through the sieve was considered silt and clay associated material. But if you do it that way, then a lot of the soluble materials will also end up in the silt plus clay fraction. The soluble materials will be washed out and are not included in this fraction.
So,
But microbial biomass, and particularly fungal hyphae, will be included in this fraction. If the microbes, the bacteria, as they grow in the soil, they produce a mucus membrane to keep them protected from desiccation. And we will get some of that material isolating in this fraction, and some of that material, and the hyphae. So the water-soluble stuff probably goes into the fractions that are washed through the
And that
sieve.
would be also dead microbial bodies too, correct?
Here,
Yes.
yes. I think that's what I mean,
Yeah.
is dead microbial bodies. So there's a lot. This is not just one kind of thing. And I think that's the take-home message from the work I've done in fractionation. Maybe I was trying too hard to give you what you wanted and tell you something interesting. The bottom line is that organic matter exists in soil in reality as a continuum. And so when we study fractions, what we're trying to do is to use laboratory methods to allow us to go in there and grab a section of that continuum. And when I say continuum, I mean a continuum of decomposition rates, a continuum of chemical characteristics, a continuum of association with soil particles, protection within aggregates, adsorption to clay alone, various sizes. So it's a mishmash of all kinds of things. We don't even really understand that much, even today, about what humus is and humic acid. We talk about it, but we don't really understand completely what it is still today. And so this research was done, and why I did this was to try to go in and isolate a fraction without chemically changing the characteristics of that fraction. So I gently dispersed it. I gently handled it from beginning to end. And what we're looking at here is something that is strictly associated with the physical structure of the soil. And I can't guarantee that it's like it was in the soil, because, you know, when you go out there and you pull something out of soil, you take a soil core and you bring it into the lab. It's different, okay? And so this is, as close as I can tell, what it was out there in nature. And that is the point of this little diagram, which I'm not sure why it got stuck. I was rearranging like crazy to try to make this clear. So what has happened through the past, however, 20, 25 years of research, people have worked on this fraction internationally in all kinds of systems, not just ag systems, is that it's pretty clear that particulate organic matter and light fraction is the first intermediate along this pathway of stabilization of fresh plant material, particularly roots, to stabilized organic matter.
But that diagram you would definitely have to show that all that is biologically driven.
It is. Then when I show a CO2 being released from this, I mean that this is where the microorganisms. I make assumptions because I know this. I make assumptions. You know, CO2 is produced because organisms are decomposing it. Organisms are oxidizing it. All along the way. Here, I've included numerous transformations along the way in numerous places. Some of this will be lost to CO2 directly. Some of this will be staved in aggregates. And if we keep going, I think we'll get to this. But I want to bring this point up too. This is a model. This is a very, very early model of the century model, which is one of the best known models for carbon cycling in the world. And I just so happened to work at the lab where this model was developed. And so the modelers got very excited when isolated this fraction, because they thought, oh, we have finally found something that's behaving like the slow soil organic matter pool. That means intermediately labile material that is got a lot of lignin, and that's one thing I didn't put on my table. That material is 50% lignin across all of the tillage scenarios, including the native.
Lignin. Which one what pool was it you said that?
This one.
Structure. Okay.
So that means it's turning over. I usually think about decades to hundreds of years. They could not figure out what they said. Well, this is probably, you know, fresh material from plants, and this is probably where the microbial biomass are, and that's where the dead biomass is, and it's cycling very rapidly. But they couldn't figure out a fraction that would represent this pool. So they got very excited about it, and it turns out that let me see if I'm saying this. The next few slides, I think, will help you to understand why it turns out that it really isn't just slow organic matter. Some of it is, and some of it cycles with the active fraction. Okay? And what I'm going to show you over the next few slides, some data, some of which was not part of a major experiment and never got published, but I was doing to help to try to gather information like a detective to design an experiment to get at this. So the first thing I did, and I'm here in Iowa now, is I went to a couple of places in Iowa, 2 field sites, and I looked at palm carbon in the fall, and I looked at palm carbon in the spring. And the first thing I noticed was that palm carbon increased over winter. So that means there are inputs into this fraction. Well, that makes sense if it's coming from roots in cropping systems. So the roots are decomposing, and the organisms that are decomposing the roots and the material that come after that are all associated with this. And so we're getting an increase in this fraction. Remember, this is not just roots. A lot of it is roots, but it's also all the microbial byproducts that are associated with the decomposition of the roots. Dead biomass and exudates that are sticky enough to stick around and not water soluble. So I said, okay, that's interesting. So then I had a graduate student from Germany look at seasonal dynamics of palm, carbon and nitrogen. And we looked at 4 different places in the cropping cycle. This is a corn-soybean system, chisel plow tillage, no tillage. And we also looked at the soybean corn aspect of that. So this is growing corn this season. This is growing soybean this season. And we followed the changes in palm across the growing season. And this really interested me because look at this. Up here where you're growing corn, basically, and there's a lot of noise in this data, but basically there's no change in the carbon content. Whereas when you're growing soybean, you're seeing a large change in carbon content in this fraction from the beginning of the season to the end of the growing season. Okay? On the other hand, with nitrogen, all of the systems lose nitrogen across the growing season from this palm. Okay? And so what this said to me was, this fraction, the decomposition of nitrogen is decoupled from or the loss of nitrogen is decoupled from the carbon loss in this. This was interesting. This work went back to Germany, and the student went on for a PhD, and it got lost in the embers of science, and the only place you can find this information now is in her dissertation. But I want you to know about it because it's important, and to my knowledge, I haven't seen anything like this published, and I'm unable to publish it, but I want you to know it, okay?
Well, what's the name of the graduate student?
Her name is Sabine Heumann.
And you can find it online.
You can go to the University of Hanover. Unfortunately, it's in German.
Oh.
That's the problem. And that's why I can't publish it, or I would have. It's in German. It was never translated. Okay, so at some point, this may get out in a different way, but it hasn't gotten out yet. So this was intriguing to me. So I said, well, something is going on really early in the growing season here that I got to check. But I want to summarize this in a different way so that you can see what's going on. So what that data says is that tillage effects were less important than the crop effect in this system. And so this totally surprised me, coming from a background of tillage is bad and no-till is good. Tillage disrupts aggregates. Tillage, then obviously keeps palm active because most of the palm is inside aggregates. In this system, we were seeing the impact of the current crop, and I would also like to say, remember, the impact of the residue is probably what we're seeing more. So in the carbon, in the soybean system, where soybeans are growing in that year, we're seeing the impact of the decomposition of the corn residue. The corn residue is decomposing and pumping carbon into the palm fraction over winter, and 60% of the carbon from that fraction is lost over winter, and 70% of the nitrogen is lost, and the C to N ratio from spring to fall change is quite dramatic. This happens because you're losing more nitrogen than you're losing carbon, okay? But if you're growing corn in a soybean residue, you're seeing no net loss of carbon from the palm. You're still seeing nitrogen loss, and you're seeing an almost identical change in C to N ratio, 11 to 20 versus 10 to 22. But this was for a completely different reason. Now, all of this can be related to net loss of nitrogen. And what I think is happening here, this doesn't make sense if you look at the C to N ratios of residues. You would think soybean residue, because its C to N residue is so much lower, would decompose much more quickly and lose much more carbon. And I think what's going on here is that, remember, it's net. You're also seeing the impact of the residue of the exudates that are being produced by the corn crop, which we know corn produces a lot of rich exudates, and those exudates are attracting a large biomass. And so we're actually getting a carbon change across the growing season as a result of exudation, decomposition of exudation, and this is a conceptual hypothesis, folks, okay? This doesn't test this, but it's one explanation, all right? And it also could be because there's turnover of fine roots in corn across the growing season. And in fact, work in the past 20 or so years has shown that there is turnover by people that study root turnover, okay? So this was interesting. Unfortunately,
So I have a question for that.
yes.
But in the soybean, where the disotrodes and all these are fixing nitrogen for the year, and you're leaking amino acids, and you mix that into the equation for the soybean, wouldn't that equate for some of the mineralization of the particular organic matter?
I'm not sure I understand your question.
Well, I guess.
Yes, there
yes.
is mineralization.
Yeah, because these lagoons are fixing.
Are
that.
producing, yeah.
Yeah, they're fixing and they're leaking, and
Yes.
they're throwing nitrogen into the system, and there's these incredible.
There's light nitrogen available for decomposition of the.
Absolutely.
Absolutely, yes, that's right. Absolutely. You're good. Okay. Say again?
She probably didn't touch that.
We didn't have corn after corn in that rotation. And I'm sorry that we didn't. I'm sorry about a lot of things, but this is stock, okay? Anyway, so the final piece of this part is I went out there one season at a single field site, and I studied the early season changes in particulate organic matter. So now we're looking at from mid-March to the first week of June. Spring here in Iowa, early spring here in Iowa. And I looked at what I called big palm and all palm, and then these are two density fractions of all palm, all right? And the thing I want you to take home from this is we're getting inputs into a fraction in the spring, but the inputs are coming into this big palm fraction, okay? That's what's causing the total palm to increase in the spring. So it's cool. We don't have a lot of material here yet. Decomposition is starting. Boom, we get a big spike in the spring, just like I'd seen at so many other field sites after this. I looked in a lot of places. But most of it is due to big palm. And by the early May, it's gone. Okay, so this, to me, says this is the cyclic release of material. This is the active part of the palm fraction. And then we have this stuff that's left over that just chugs along and turns over decades. All right, this is just one little study for 1 year, but it fits the picture. And so the other thing you need to know is that this larger palm is less dense, less decomposed, and has a wider C to N ratio over than the smaller palm. Smaller palm is associated with mineral particles. Smaller palm has a narrower C to N ratio, and this is retained across growing seasons. This stuff pretty much floats free in the soil. This stuff is protected within aggregates, okay?
So
No.
which was in the water, so the active coal would be in the water solution, as it be picking it up. The active, right? Would you have some of that? Which
No,
parts?
no. And interestingly, because I wasn't able to separate the active from the silt and the clay, my purposes for these experiments were not the same. The active pool's effects are diluted by the high amount of carbon and nitrogen. Remember, 60% of the carbon and nitrogen, even in a native system, and up to 80% in the tilled systems, are in the silt and clay, which are highly recalcitrant. And when you include this small amount of very highly available water-soluble stuff in with this big swimming pool full of very narrow, not decomposable stuff, you're not going to be able to tease apart the differences.
Okay, so
but it's in there.
Okay, so what you're saying is most of the stuff you're doing is it's embedded in the fractions of the minerals, and that's what you're dealing with?
This stuff is, no, this is not embedded in the fractions of the minerals. This is the stuff that is released when you add a hexametaphosphate dispersant to soil, which basically blows apart the macroaggregate structure. It blows apart the aggregate structure. And so you're left with this material and silt and clay particles, and also the soluble stuff goes through the sieve.
So,
Okay.
one more question, I'm sorry.
Yeah.
But
It's okay.
I'm learning a lot here.
I'm
So,
sorry I didn't
what
plan
you're
it more
saying,
to
so
teach more.
You have this incredible aggregate, you blew up the aggregate, but it does not account for the stuff that actually floats in the water system that's floating around.
Oh,
also.
like the very less dense stuff?
Yes.
The less dense. That would be in this fraction, but it would be, once again, a subfraction, because the stuff that floats on water has a density of 1. And this material has a density of between 1.6 and 1.8. Okay? See, there's so much. All right, now I want to get to this. So a graduate student and I decided I wanted to work on trying to definitively show the source of palm coming from roots rather than shoots. And I'm going to go through, this is a very complex experiment. I'm going to go through it really fast because I do want to get to the end. But what we did is we grew up oats, and we chose oats because we were trying to couple it with Tom Casper's cover crop work, and because we were trying to get some handle on cover crop storage. And we grew oats up in a in a big growth chamber, and we pulse labeled them as they were growing with carbon-14 CO2. And we did that, and we did some with labeled, and we did some that weren't labeled, so that then we could cut off the residue from the labeled stuff, the surface label, and put it on the soil that wasn't labeled. So we have labeled shoots and unlabeled roots, and vice versa. We have labeled roots in the other pot with unlabeled shoots. And we spread it across the top, and then we put a CO2 trap in here to trap all the C14, and we let it decompose for an entire year. And we sampled in the growth chamber, and we sampled it every 3 months. Totally disassembled this big pot, sampled a whole bunch of fractions, including the CO2, and then we did budgets at the end of the time. Okay, so that's the short version of this. And here is what we found. Okay, we found that if in the C14, if the shoots were labeled with C14 and we tracked the movement of carbon into soil from shoots, only about 10 or 11% of the shoot carbon made it into the soil. If you're practicing this, is meant to simulate no-till. Almost 70% of it was lost off the top as CO2, and there was very little in the palm fraction. Okay? If you labeled the roots and then allowed that to decompose and look for movement, there was significantly less carbon lost as CO2, and here's the big punch, a lot of carbon stored in the soil, almost 40%. And a good chunk of it is in the palm. Okay? So this says that finally we have a tracer that allowed us to follow the movement of carbon, one carbon molecule at a time, basically, from a shoot or a root into the soil pools. And this definitively established the importance of roots in the turnover of carbon in no-till. And I also want to say that at this same time, I was working with Don Rakowski on a study where we were looking at carbon loss after 35 years in silage corn. And I worked with him on this study, and it was published in the Journal of Soil and Water Conservation Society many years ago. And we were very surprised to find out that after 30 years of silage corn, there was absolutely no difference in the carbon content in the surface soils in the silage corn versus the system where the residue was returned. All right? And so, not to say that there wasn't carbon lost over 35 years, but just that there wasn't any difference in the carbon content when you removed the silage or you didn't. And so that, at the same time, these two studies really supported each other. So we're seeing it in the field too, even with tillage. And I would say tillage, because when you till the soil, you're speeding up the decomposition of stuff like palm, you're going to lose even more carbon dioxide out of the top. And that was the question that someone asked me when I did this study. And since then, the Canadians have actually done a study that shows that. And I don't have the link here, or the study here, but I can show it to you. All right, let's get into the end of this, because I want to say a few things about what I'm doing now, and I appreciate your patience. So I talk about soil health all the time, not as much as Ray does. I don't think I'd survive if I traveled as much as you. But I came, what used to be soil quality has morphed into soil health, and I think that's good because it emphasizes the importance of soil biology. And you've learned and heard an awful lot about soil biology, I'm assuming, in the past couple of days. But before that sorted out, I had to develop a way, and we were in the interim stages, I had to develop a way to talk about soil quality and soil health, and I thought that there was it was useful to separate them, because it helps you to understand that there are different things going on in soil, and it's complex and interrelated. And so the soil quality effort really focused on measuring soil properties, and so that's like the framework. What's there in the soil? What do we have to work with? And the soil health concept was really more about the soil biology. So what are they doing? How are they cycling nutrients? How are they storing organic matter? How are they helping to build and degrade aggregates? And that really emphasizes that everything that goes on in organic matter in the soil is as a result of the activity of organisms. And that what this focuses on is, well, maybe I better start looking at soil function, okay? Because we have a lot of data here, and we've got a lot of awareness and a building database now, especially with the increased interest and awareness of soil biology. Pretty soon we're going to have as big a database here as we have here, and we're going to start wanting to say, what does soil function do? So this is just a little diagram that probably just, this is soil quality looking at integrative indicators and soil properties. And I'm going to tell you a little bit about the work I've been doing in organic for the past 15, 16, almost 18 years. First, I started working at a field site just outside of Greenfield, Iowa, with an Iowa State collaborator. This is a long-term study. It started in 1998, and it was certified organic in 2000. The cropping treatments are a three crop rotation, corn, soybeans, oat, alfalfa, and a 4-crop rotation, corn, soybeans, oat, alfalfa, alfalfa. And we compared this head-on. Don't worry about this, because this rotation has changed a couple of times. There's one rotation that's changed a couple of times. I can't see this very well without my glasses on. Anyway, it's the bottom. Focus on the top 3. And we also have embedded in this completely randomized design done on large plots, production agriculture size machinery goes out here, with a corn soybean rotation managed conventionally, as it is in Iowa. We use composted swine hoop house manure or poultry as a nutrient source for organic corn at this rate, and also to the oats. The oats got less than the corn, and conventional got this. What I want to talk about is, ever since 1998, I've been going out there every fall and taking cores to follow the change in soil to a depth of 15 centimeters. And we don't have to talk about this. So here's what I find out. This is as of fall of 2012. I haven't gotten the 2013 data in a graph yet or a table, but this is the take-home message. Even with all of the extra cultivation in the organic systems, because it's disked in the spring and then it's rotary hoed and then it's disked a number of times to control weeds, whereas the conventional gets one, we have seen almost since the first or second year, I think even in the first year we were seeing a beginning of it, higher soil organic carbon in the organic systems, particularly in the 4-year rotation compared to the conventional. More total nitrogen, more POM, and in this year the microbial biomass was not significantly different, but last year it was, and POM wasn't. So when you have a long-term record like this, you can see that, that active fraction, it really depends on the climate and what's going on, and the pattern of rainfall and precip, I think, and what's going on with the crops. The thing that's really important to notice here is that the ability of this soil, as measured by nitrogen mineralization potential, has been significantly higher in the organic system probably for the past 12 years. I didn't bring all the graphs, but it is significantly higher. That means that this system has developed the ability to supply nitrogen without external fertilizer. The other thing that you want to know is that phosphorus, potassium, and magnesium and calcium, macronutrients, and this is the same thing for magnesium. Most years it's significant. I don't know why this didn't turn out significant this year. 2012 was a weird year. It was dry. Who knows? But so there's not only more nitrogen, there's also higher cations. There's higher P. So CEC is better. The nutrient supplying capacity of the organic soils is higher, and the acidity is lower because we've stopped adding chemical fertilizer and we've stopped pumping hydrogen ions into the soil. So we're increasing it. All at the same time, we're not seeing any difference in bulk density, at least not in the 15 centimeters, top 15 centimeters. This is a lot of work to do this, so I really didn't look at anything but the surface soils over long term.
This was chicken manure?
Poultry manure over the past probably 4 to 5 years. Prior to that, it was swine hoop house manure, which means the manure was pulled, had a relatively high C to N ratio. It was basically the bedding from overwinter in the hoop houses. Okay, so a slight change.
Of that chicken litter they were putting on?
They were, say again?
You know the race?
Yes, 134 kilograms of nitrogen per hectare.
Which equates to how many tons?
I don't know. A lot less than the swine hoop house because the chicken manure is hotter.
So Cindy, the conventional was chemical fertilizer, right?
Yes, urea.
Because what this is showing me is how important carbon is for the system, for food,
Yes.
manures, and rotations,
Yes.
and the plants.
Yes, it's very important right in here. Okay, and it's excited me, and I know I picked the far range. I didn't tell you why I went to organic. I picked what I thought would be, we had the extended rotations, we had the cover crop in this system. Rye is planted after corn every year in these plots, and so we had it all. I thought we would have the best possible chance, and we're adding compost. So I'm looking at the best possible scenario. The only thing that was not optimal was the fact that there was so much tillage. And it turns out that because we're feeding the organisms, we're actually overriding the impact of this tillage and still allowing the system to build organic matter. And I put this one graph in here just to show you the kind of data that we have over long term. So not only can you see what's going on in a given year between the 3 cropping systems, You can also look at the long-term pattern. This is for total nitrogen. And this shows very clearly the total nitrogen is going up in this system, and the difference between the conventional, which is bright blue, and the organic, which is red and green, the difference is getting greater and greater the longer this stays in organic. I also want to say that yields at this particular site, unlike a lot of sites, have been equal to or better than the conventional treatment for nearly every year of this experiment. And we've been criticized for that too. You must be doing something different. You're putting on too much compost, blah, 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 blah. But the truth is that in these highly productive Iowa soils, we're still able to grow good crops and increase soil quality or soil health because of the change in organic carbon in the organisms over time, and maintain yields. The 1 year we didn't have so good was 2012. I also am working on organic no-till, and I could talk about that at some other point in time. I can tell you that what I do know at this point, and its study's been going on for quite about 6 years, is that microbial biomass is bigger, greater under no-till. Still no change in carbon compared to organic conventional till and organic no-till. The other thing that's important is bulk density is reduced in the organic no-till compared to the conventional. And aggregate, there are more macro aggregates in the no-till than the organic. So what that says to me is that if we can solve the problems associated with getting good yields with organic no-till, we've been getting tremendous soybean yield compared to, and this is similar to other people looking at organic no-till. Soybeans do very well, but corn is tough. The only place that does a good job with corn is Rodale, and we're all convinced there's something magical, not magical, but something unique about the soil system at Rodale. This is the study that came out of that other study. So I said, we got all this good stuff going on with soil properties and soil health and soil biology. What about function? So function, CO2 flux, yield, infiltration rates, leaching, nutrient use efficiency. So what I thought would be an ideal way to look at this in this particular place in the country was to look at tile drain leaching of nitrate under organic systems compared to conventional systems. And we had to get the money to actually instrument this site and install individual tile drains that isolated the tile drainage from each individual plot, which are also production size, just like the site in Greenfield. This is the experiment that I wanted to show you at the Boone Farm yesterday. So here it is. And we've got, I took from that experiment, well, here's a picture of the tile installation. We selected this field. I found it early on, and we kept it in oats and alfalfa for a number of years. I think it was like a total of 5 or 6 years until we could get the funding to put the tiles in. So no chemicals applied, but no real crops. So we were building soil, or trying to. No chemicals since 2006. We installed the tiles in 2011. First year of cropping was 2012, the worst year we'd had in who knows how many years. Here's the setup. It's set up in 3 tiers. The tile drains run down the center of each plot, and one sump collects drainage from each of 3 plots, and so there's 10 sumps lined up along the edge of the field here. We have 30-foot rows between the tiers to help to partially satisfy organic certification standards, because we really wanted to certify this site, but ended up having it be not big enough to actually officially certify this site for organic food production without sacrificing having enough plots to get replicated data, because tile drainage flow is very variable in the field. And so we made a trade-off. We put alleys here, there's alleys here, but between the plots, there's an apron that goes down to, I don't have it written here, I think it's 8 feet, between to block any kind of lateral flow, so a plastic apron. All right. Here's a picture of the site. The rotation that I chose to put on this site was the 4-year, because that was the rotation that showed the best nitrogen improvement and along with carbon improvement. So I figured we had only room to put one treatment. I want it replicated with each crop on the ground every single year. We wanted to have a conventional in that as well, because we want that direct comparison, and looking ahead to integrated cropping systems with livestock, because many organic farmers integrate. We included a pasture hay treatment in a replicated fashion too, thinking down the pike in the long term. Okay, there are 5 replicates per system. That's a lot of land, because there's variability on this site. There's a slight slope, so there are 3 different soil types here, and the drainage is very different. All right, and I could spend an hour talking to you about that. I want to tell you that we have a weather station on site, so we not only get continuous tile flow monitoring here to look down into the pit, so we get real-time flow monitoring. It's wired into this weather station and can be downloaded. Well, we had to make a decision, Ray. We had 30 plots, and I went through every possible scenario, including why the heck didn't you include a conventional 4-year rotation in with this? And because the money to fund this came from an integrated call for organic water quality, we wanted to look at organic corn, soybean, oats, and alfalfa, and we didn't have enough data on our no-till systems yet to actually put this in place. This is still what is the predominant system in Iowa. Okay? Yeah, dreaming, maybe after we have 8 to 10 years of data on this site for tilled, maybe we can transition it to no-till. With so few plots, I had to make a decision. I wanted to include a 3-year rotation and get a wheat crop in. That was another study I worked on for a number of years with folks here at the Tilth Lab. Corn, soybean, wheat, red clover. I wanted to do that and couldn't do that in this and put 5 reps out there. So this was decided based on the limitation of the site and based on the best guess that we could take. And this is frequently what happens. It really is. And this, believe it or not, is going to provide some unique information for this area.
I guess for myself.
And we're still trying to get money. We haven't been successful for a number of years now in getting continuing funding for the no-till, organic no-till part. And I think it's because competition for that pool of money has gone up, and they're spreading the money out to other people. But we're trying again this year to the organic call. If we get funding, then we'll see. Maybe we'll put in some
Have you thought
lysimeters.
about CIG?
Oh yeah, we've thought about all kinds of things. The problem is I've spent the past 6 months doing nothing but writing grants, and I need to look at data. And this is true. It's pretty much true. To look at data because I need to keep this site funded. We ran out of money last year on this site, and we need to keep it funded and running. So there's a lot of opportunities. I've looked at CIG. I'm putting in an AFRI grant this year to try to expand the microbial aspects of these plots. But for right now, believe it or not, this data will be unique. This hasn't really been done. It's a ton of work, and it took a lot of money. It took the entire first year's budget to install the infrastructure. And then it took the rest of the time to get the plot started. So what I want to do in this last slide is to give you a flavor of the kind of data. Yields, I have a slide for yields. These 2 years were very different. In 2012, we had the same amount of rainfall total as we had in 2013, but in 2012, we had a mostly dry year with very little. It was like 23 inches of rainfall. In 2013, we had a really wet spring, and then we had nothing. And so all the rain fell in the spring, and so we had a lot of problems in 2013. We had good yields in 2012. The organic and the conventional yielded the same for both corn and soybean, but in 2012 we had a lot of problems and the yields were very depressed. And the organic didn't do very well because there wasn't enough nitrogen there, because we got the compost down early in the season before the rain started, and then we couldn't get back out into the field until the rain stopped and the nitrogen was gone, and we tried to save it and we couldn't. Take home from here is corn. This is nitrate N loading. This means kilograms of nitrogen per hectare leaving in the tile drains. Organic corn, 10.3. Conventional corn soybean, 10.1. Organic soybean, 8.8. Organic oats, dramatically reduced it down. Pasture forage, I was surprised, but the forage crop was very young at this point. And there are some legumes in that rotation. Organic soybean, corn and soybean, about the same. Together here with the conventional corn. Oh, that's why I put that there. To show you that corn and soybean aren't going to change this rotation. They're going to probably be the same, but I'm not sure that'll hold out for all seasons. What's going to make the difference here is the presence of a small grain and the presence of an extended rotation to reduce the total impact of nitrogen leaving the tile drains. So the years that the oats are on the ground are going to make a difference. And as we move into the season where we have maybe halfway decent rainfall in the right time in the right place, we might be able to give you more data. The other thing is 2012 was the first year, and the tile, the problem with the disruption of the soil from the tile installation isn't over yet. This year will actually be the first year, according to my expert on tiles, Dan Janes, in this lab, that we can actually truly trust that our data isn't going to be impacted as much from the tile installation as from the cropping system. So what we found is not only are we seeing that long-term grain rotations in Iowa are stable and resilient and improve health, there is a promise, albeit we don't have enough data yet, but I like to be optimistic, just like Jerry. There is a promise that these rotations managed organically in Iowa will improve water quality in the tile drains and exiting to the surface waters, which is a big problem for us here.
You make that comment.
I probably should have not made it so broad. I get exciting. At this one site, it's stable. This site is up on a ridge, and there isn't any soil loss from these systems in terms of the corn and the organic systems. We keep a cover crop on the ground, and there isn't soil loss. But if you have something that's on a sloping system like this new site, we're probably going to see problems with erosion. And I'm trying to get Martin Chipotello or Chipoletto, you say his last name, Tom, Chipotello, I'm sorry, Martin, to work with us out there and look at erosion from the various plots, but he's busy doing so many things here and getting his program going. But yes, erosion is a problem, and that's, I think, where prudent management of cover crops and increasing the time that the soil is covered with some sort of residue or growing plant is going to help. So organic management doesn't solve all the problems, but it does help to solve some of the problems associated with conventional agriculture here. So thank you for your patience, and I appreciated the opportunity to talk with you, and I hope you have a good rest of your day.
Thank you.
Aaron.